Hello, good evening, everybody. All right. Hello. I hope you can hear me loud and clear. This is going to be the last lecture on the session on seminology. And this lecture, I'm sure, had already been posted. This is the last of the lectures on the structure of the sperm. We've done the normal structure. We're going to do the abnormal structure today. Tomorrow, God willing, we're going to start semen analysis. All right. So take note. All these things are supposed to help you to appreciate the semen analysis in its proper clinical context. All right. So all the andrology and then the spermatogenesis and everything is supposed to help you to appreciate the semen analysis more properly scientifically. All right. So as we move into the semen analysis, I'm sure a lot of things, I'll just mention them, hoping that you already have the basic understanding of it. All right. So today, like I've said, it's going to be very brief, and it's also going to be just like the morphology of abnormal sperms. All right. I'm sure it's been posted already, and I'm sure some of you may have gotten the opportunity even to go through already. If there's something from previous lectures that you have some challenge with any aspect of it, please, this is the time to bring it out. And then I can try to explain as much as possible. If there's nothing worrying, then we move on. Now, the previous lectures on the seminology has been put in session 1, session 2, session 3, and session 4. All right. Some aspect may overlap. For example, if you, for a particular lecture period, some of the things that we discuss, you think that may spill over into the PowerPoint presentations, but I'm sure you understand, because just like sometimes just a repetition. All right. I want you to understand the entire concept more properly. So if you read the lecture notes and then the video presentations, and there are some abnormality, no, not too much of abnormalities, but if there is some overlap, I'm sure you appreciate it. All right. Now, if there's no question from the previous discussions, then we move on. This is, like I said, going to be the last session on the seminology. And it is sperm morphologic abnormalities. Now, we've seen how the sperm is developed from the germ cell to the spermatogonia, to the spermatocytes, to the spermatids, and for it to be spermiated. That's spermiation, where it's now leads into the tubule, and how it is transported from the tubule to the red testis, to the vas efferens, through to the head of the epididymis, through the corpus, through the cordia, and then now be stored over there. You've seen all the morphological and maturational changes that will take place also in the epididymis. We've also seen the role the epididymis plays in protecting the spermatozoa. And now we want to see this spermatozoa, for that matter, this sperm. How does it look like? We looked at the normal morphology, the head, the neck, and then the tail. We've looked at all aspects of it. And we know the ideal characteristics, how the head should look like, how the neck should look like, how the tail should also look like. That is the ideal characteristics or features. Now you have to know it very properly, because if you are doing the semen analysis too, you are going to look at the morphologies. And we have what we call the Kruger strict criteria. Kruger strict criteria wants to say that if a sperm is supposed to be normal, then every aspect of it, from the head to tail, every aspect of it should conform to the ideal characteristics that we have mentioned earlier. Okay, and the overhead and every so if there's the least deviation of it, it's supposed to be an abnormality. Okay, that is the strictest sense of what we call the Kruger criteria. All right, so a lot of time people are confusing this Kruger criteria with a certain criteria. Of course, that was also established by WHO some years back, where morphology was not all that strict. So, if the head is okay, and even there's a little problem with the tail and this thing, for as long as the head is okay, let's pass it. If everything's okay, except that there was a small, for example, you said that the cytoplasmic droplet is a normality, it's a normal feature of sperm development. But where should it be? It should be somewhere around the neck. Sometimes you will find this droplet at the tail end of the tail, so we call it terminal droplet. As you move along, so to some extent, if everything is normal and just this small droplet at the tail end, just pass it on. Understand? But Kruger criteria will not. Any least abnormality is considered an abnormality. In that case, it says like the fewer the merrier. So even in that case, even if you have just 4% of the entire population of sperms being normal, then the whole semen parameters is considered to be normal. We haven't got to the semen analysis. What I'm saying is that you should know the sperm morphology so well, so that when it comes to the morphological assessment during semen analysis, the evaluation of the morphology, then you are going to use this information here to help you to now know how to write your results. And so over there, you see that, probably we are using the latest Kruger standard, which is saying that if you're able to get 4% of the sperm population to be normal according to the Kruger strict criteria, then the entire sperm parameter with respect to morphology is also normal. I hope that you are getting my point. Because it's saying that every smallest deviation from the normality is going to be considered as abnormal. So that if you have 4% to be normal, that 4% means that it means that there could even be some more cells which in some way or the other, they are not too much an abnormality. But because you're using the Kruger strict criteria, you consider it as abnormality. So it means that the 4% is a lot. All right. So let us now look at the morphologic abnormalities and look at it, and so that once you are doing your semen analysis, when it comes to assessment of morphology, it is this information that you are going to use it in that direction. I hope that you've gotten it. So sperm morphologic abnormalities. Poor sperm development can lead to abnormal sperm morphology or poor shape. That point is clear. As we said, look at the processes that the sperm goes through from the germ cell to the spermatogonium and blah, blah, blah. At a point, it may be considered to be apoptotic, because once the abnormalities are even too much, the body itself will target as apoptotic sperm, and then the cytosol cells will deal with it. because the concept is that Sertoli cells will have to deal with all abnormal apoptotic cells, and then even their cells, and then the cytoplasmic droplet, the Sertoli cells will have to deal with it. But sometimes some of them will push through, will pass through, because they are new. We said we have over millions of sperms are produced per day. So yes, you are likely to have some of the abnormal cells which are supposed to have been picked up as apoptotic cells to have been ejaculated. Once they are ejaculated, then the scientist who is looking at the sperms will not have don't know these ones, they are not normal. Once they are not normal, it is unlikely that they will be successful in what you want them to do. Finally, what you want them to do is to be able to go through the harsh vaginal environment and then be able to okay. Please, somebody is not hearing me. Is it the same everywhere, please? If somebody says, all that I'm saying is that we are not doing semen analysis now, but we are doing the sperm morphology. The sperm morphology is going to be part of the parameters to be assessed during semen analysis. And it is important to deal with here because it is part of the sperm development. You get them to conform to set shape. You expect them to come a certain size. You expect them to get a certain appearance. And this is what goes into morphology. Morphology is nothing, the structure, the shape, or the form, or the appearance of a spermatozoa. All right. So, and we know that we have three sections, the head, saying that we need to know this thing because when it comes to semen analysis, there's a parameter on morphology. And morphological parameter is assessed based on what the structure is supposed to be. All right. So if you know what the structure is supposed to be, then if you are going strictly by that structure, it is called the Kruger strict criteria, that the head should be this way, exactly that. If there's any small deviation of it, it is classified as abnormal. The neck should be this way. If there's any least deviation of it, it is abnormal. The tail should be this way. Any least deviation of it is abnormal. All right. So if you consider all of them to be abnormal, then those which will go by the ideal characteristics should not be more than 4%. So it means that under normal circumstances, 4% of anything should be a failure. But if it comes to morphology and you are going by the strict criteria, 4% is supposed to be normal. Because it means that you have ruled out even those who were normal, except some small problem with it, you consider it as abnormal. All right, in that case, then the percentage, the 4% percentage is considered to be very, very, very acceptable. All right. So we're looking at the abnormalities. We look at abnormalities in the head region, looking at abnormalities in the midpiece, abnormalities in the tail. So we call them head defects, midpiece defects, tail defects. All right. The medical term for when semen sample has a large number of abnormally shaped sperm is called teratozoospermia. or teratospermia. Now, let me take my time to explain these two concepts very well. Teratozoospermia and teratospermia. Now, the word terato is got to do with a monster. All right. So terato got to do with monster. And in your opinion, what does a monster look like? Something monstrous is like a chimpanzee. A certain type of chimpanzee is put into the monster group. It's a monster. It's like it has a very bad shape. I mean, it is not that it doesn't got a pleasant appearance. All right. So once you're able to connect terato with a monster, then you are looking at a weird shape. Okay, right. So, and the word zoo is, of course, about animals. So, and you know that within the semen, all right, you have fluid portion and you have the cells or the animal section. The animals are the sperms. I just want to take these things so that you can appreciate them properly. So the zoo is talking about the animals in the entire semen, and the animals in the semen are the sperms. So teratozoo, you are talking about the shape of the what do you call it, of the sperms. In the spermia is the entire semen. Okay, the spermia is the entire semen. So we are talking about the shape of the animals in the entire semen. Okay. Now, what about teratospermia? Sometimes it's like everybody knows that semen contains sperms. And if you talk about the shape, you're talking about the sperms in there. So it's like, even if you don't say sperms in the semen, I mean, automatically it is what it is. So it means that if you bring out the zoo, if you take up the zoo, it doesn't change anything. That's what some people believe. And so instead of saying teratospermia, it is also acceptable to say teratospermia. But let me take you back. Let me take you back. So in that case, it means that if you are talking about azoospermia, which is A means absent, okay? Lack of. Zoo is the animals. Zoospermia. So azoospermia will also mean no sperms in the semen, okay? Azoospermia. If you are saying that, oh, then it is also normal to take away the zoo there, okay? Instead of saying azoospermia, you want to make it aspermia, gives you a different meaning altogether. Aspermia means no ejaculation. Ejaculation, absence of semen, absence of nothing coming out. Remember, when you thought that in the retrograde ejaculation, it can be total retrograde ejaculation, where now total retrograde ejaculation, everything is going into the bladder, and so nothing will come out. So the person will feel orgasm, yet there's nothing coming out. That is spermia. But so that take the note of this one very well, because sometimes it becomes very complicated. For example, if you say oligozoospermia, and then you say the same thing as oligospermia, it means that you're taking off the zoo. Okay. Now, so in this particular one, if you say azoospermia, if you took off the zoo, it becomes aspermia. Aspermia, the meaning of aspermia is different from the meaning of azoospermia. I don't know whether you are getting my point. All right. So I just want to say that teratospermia, you could take off the zoo and then make it teratospermia. When it comes to other things, oligospermia, and then it is also the same thing as oligozoospermia. They've taken off the zoo to make it oligospermia, so forth and so on. So let's take this note as we move to semen analysis, you understand some of these things properly. But now we're talking about the shape, abnormal shapes. I'm saying that is the abnormal shape, that medical terminology for abnormally shaped sperms are called teratozoospermia, which could take off the zoo and make it teratospermia. And I'm saying that the zoo there is actually talking about the sperms in the semen. the sperms in the semen. This is the zoo. That the sperms in there. That's why when you say azoospermia, A means, medical terminologies. When you talk the prefix A or an, it means absence or lack of. So azo, so absence of sperms, absence of zoo, lack of the animals, azo, in the entire semen. So that is why how it becomes where there's no sperms in the semen, it is azoospermia. Here the terato, and I'm saying that terato got to do with monsters, a monster, a monkey, I mean some chimpanzee type of thing, and it's talking about their shape. Because sometimes once we move along, we're going to talk about actinozoospermia. Okay. Sometimes so people confuse actino and then terato. Which one is talking about the shape and which one is talking about the movement? The actino is talking about the movement, motility. And then terato is talking about the shape, morphology. But sometimes, like all of us know, sometimes you can get confused. The actino and then the terato, which of them is about the shape? Which one of them is about the motility? Once you remember that I've told you that terato is got to do with a monster. And a monster here, we're talking about how, I'm not sure monsters are beautiful, how bizarre shape they have. Then you can, terato is got to do with the shape because it's about a monster and they don't have a nice face. All right. Right, so we move on. Still on sperm morphological abnormalities. Right. So firstly, talking about the head defects. The common abnormalities in the sperm head include macrocephaly. Macro, all of us knows about large or big. Cephaly is talking about head. Cephalic. So that is what it is. So large head. Now, of course, micro is smaller. So microcephaly becomes small head. Then you have a pinhead. Pinhead, all of us know about the pin. Look at a pin, safety pin, or anything which is a pin. You see that the head is like a small dot on the entire sharp portion. So a pinhead is almost like it doesn't have, the head is supposed to be an oval shaped thing sitting on the neck. All right. If this head is now like a pin, the head is like something small, then it becomes a pin. It has a head, but the head is so small that it makes it look like a pin. So that's called a pin head. Remember, there could be a situation where even that small pin is not even there at all. That becomes headless. So there's a difference between pinhead and headless. Sometimes people say that headless is the extreme example of a pinhead. All right. But a pinhead means that it has a head, except that it's so small that it's like a pin. I mean, something small on it. That's a pinhead. Headless is headless. You're only seeing the tail, the tail of the spermatozoa. All right. And with nothing there. You also call it decapitated. Cap has got to do with head. And then D means remover. I'm sure all of us know that. The medical terminology for D means remover. I'm sure a particular example is dehemoglobinization. Okay. You know that hemoglobinization or hemoglobin is a moving of the hemoglobin, dehemoglobinization. So D is usually a prefix standing for removal, deamination, removing the amine acid, amine component, deamination. And there are a lot of examples of D. So decapitated means removing of the head. So it's the same thing as headless. All right. Head is like when something tapers out. It's like how do you call it? Like a pencil of a sort. The head is oval. It takes the shape of a pencil. All right. And then that shape is not typical oval shape. All right. So we call it tapering or tapered head. The thin, narrowed head. So a thin, narrow head, just like we said that headless is an extreme example of thin head. The same way, the thin, narrow head is also an extreme example of the tapered head. The tapered head, I've told you, is like how do you call it? A pencil, how a pencil is shaped. Of course, not as long as that, the structure of a pencil. And then it has now become much smaller. All right, so that becomes thin, narrow head. That's an example of the tapered head. Then we talked about the global zoospermia before. Global zoospermia, when we talked about globe. And remember when you talk about the spermiogenesis, in spermiogenesis, the maturation stage where the round spermatid will now configure to give you the elongated spermatid. We said that the nucleus will move to the centric side, one side, and then the Golgi bodies will go and cap it. And there's a protein DPY which will form like a link to cover that protein is not there. Then the entire shape will not take that over shape. Then it will be rounded. Remember that, right? So that is a globe. And we said a globe, all of us have good examples of a globe, even from primary schools, where if you go to a headmaster's or head teacher's office, there's always a globe around something with a word map on it. So that is just it, global zoospermia. So that's a round-headed sperm. All right. Of course, a double head. Okay. Double head is a double head, where instead of you having a single head, it is double. All right. Elongated. Elongated is a form of the tapered head. All right. Very long. And then piriform is like a pear shape. When something has a pear shape, it's like um, this example of a syringe, not the syringe, not the one that we use in the labs. And I don't know how the local one there they use Bentua, the one that they used to what is to pass the NMS. If those of us who are born in the villages, if you have feverish, your mother will not think that you have malaria. Or south is here. These days, because of how the development, I mean, some of you have no, yeah, some of that, you don't know what I'm talking about. But for piriform, it's like a pear. If you have a pear, that the top is rounded and the down becomes like that shape is called piriform. So, but we are saying that normal hair should be oval, not a pear shape. A pear is not oval. I hope that a typical oval shaped material is an egg. But pear cannot be said to, I'm sure all of us know about a pear. Pear cannot be said to be oval, looking at the shape of a pear. So that is a piriform shape, constricted. Of course, the thing is elongated, but then the middle portion is gone inside, become constricted. Okay, and amorphous. Amorphous means that you know that the thing should be oval and smooth, but if it is not oval but rough, sorry, if it's not, even if it's oval, but the edge is not smooth but roughened, then it becomes amorphous. All right, so all these types are available. You can see them. All right, and this is a typical schematic picture of some. So A here, let me see if I can get you there. Yes, the A here, of course, this is double head. B here, this of course amorphous head. C here, of course, the round head, and of course the global zoospermia. And then the D here, of course, is an enlarged and for that matter, macrocephalic. And then E here is tapered. All right, like a pencil shape. All right. And then F is a pinhead. So it is just like there's supposed to be a head here, which you can hardly see. So that's the pinhead. That is, of course, constricted. So these are, and you can, if you have a good stained slide of sperms, you can see all these types of abnormalities from the head region. Okay, right. So we move to the more details on some of the examples that you've given. So macrocephalic or macrocephaly refers to sperm with large or giant head. All right, so macrosperm often carry extra chromosomes and may be caused by homozygous mutation of the aurora kinase C gene. What all that we want to say here is that if you have a sperm, somebody's sperm is a lot of macrocephaly, it is likely that that person, the offspring, can also be producing macro or giant headed sperm because it is genetic. Okay, that's all that we're saying there. Because some of the morphologies, you realize that, oh, probably if the sperm was exposed to, let's say, some heat, and for that matter, it couldn't give you the right shape. Then if the person is now advised in a manner that he abstains from or he avoids extreme exposure of the testicles to heat, then the sperm should be normal. Okay, because this is not genetic. But when it is macrocephalic, because it is it's an example of an extra chromosomes, which is genetic, then irrespective of whatever you do, a male offspring of that individual will also be having giant-headed sperms. That's all that you want to say. How that you understand me? All right. So this means that this kind of sperm abnormality may be genetic, and fathers may be able to pass the condition on to their sons. All right. If all of the cephalic sperm syndrome, macrocephalic sperm syndrome, because sometimes occasionally you see an occasional giant headed sperm, 1, 2, I mean, just occasionally. That you cannot say that is macrocephalic sperm syndrome. Could be just one occasional one. In that 1, 2, it is unlikely to be the genetic one, because in the genetic one, if it's genetic, then you expect that almost all the sperms will have that giant head, and of course that will be macrocephalic sperm syndrome. That could be due to genetic mutation. All right. Now, but if you have occasional 1B plus other abnormal cells, then that could not be macrocephalic sperm syndrome. I hope you are getting my point. Thank you very much. So I think I showed you this example the other time. So this large head, if you look at the normal size and look at this one, see that this giant headed or large headed, and this macrocephalic. And this is an old slide that I showed you the other time, so you know about it. This macrocephalic. So say normal versus macrocephalic. So you can see the two examples yourself. Left side, normal shape, headed, and then the right side would be the large headed sperms, macrocephalic. The micro, of course, becomes smaller. Micro, smaller. So this is the head of the sperm is abnormally smaller than usual. And you have the sizes to be less than 2.5 in width and 3.5 in length. Acrosome abnormalities are common in this sperm. And you know the acrosome should be between 40 to 70% of the head. You know this already from the normal characteristics of the normal sperm. Right. So for as long as the sperm is already small, then the acrosome will also have a challenge. You can have some without acrosome at all. We call it acrosomeless, of course. It sounds funny, but that is what it is. Acrosomeless. No acrosome. And that's usually where the head is already small. If the head is already small, it is occupied um, by the by the nucleus. There's no space for the acrosome. All right. The pinhead, like I said, pinhead is a variation of the small head sperm. Like I said earlier, in the in the in the example of the small head, this is small head, but even smaller than a small. So it becomes like a pinhead sperm. So we say that it is a variation of the small head, and for that matter, the microcephaly. All right. In which the head appears as a pin with minimal to no paternal DNA, meaning that the head is so small that most probably it doesn't even have the DNA at all. And the DNA is the nuclear chromatin. Okay. And it may not even be there at all. Right. Then the last point, the presence of pinhead sperm may point to a diabetic condition. It may or may not be usually the case. The diabetics are known to have a lot of pinhead sperms. Like I said, headless. So we had the microcephaly, then we have the pinhead, which is smaller than smaller than the pinhead. Sorry, the pinhead is smaller than the microcephaly. And then we are having a situation where even that small pin, even not there at all, so it becomes headless. Now, remember we said that in medical terminologies, the prefix A means absence or no or nothing. So, and we said that cephalic or cephaly is talking about the head. So acephaly means the absence of head. So it's the same thing as headless. And then the same thing here. So decapitated, removal of the head. So either of them means the same thing. Decapitated sperm, acephalic sperm, headless sperm, they are the same. And over here, you see them as just the what do you call it? The tail, like a thread here, and you can hardly see the head. This is even no head. This probably the head is just gone, decapitated. All right. So in this case, because they don't have a head, definitely they have no genetic material. Okay. This type of sperm may look like a pinhead sperm condition and give the sperm appearance a loose piece of a string, as I've tried to explain already. In this case, do you expect this kind of sperm to be able to fertilize? Because fertilization will require the head to have a nuclear listing, to have acrosome to be listening. So if you have sperms that are full of this one, of course, yes, you have sperms, but you cannot fertilize. All right. Good. Then the tapered. So the tapered head has a cigar shape. So either a cigarette shape or like I've said, I've tried to use the pencil is more, I mean, everybody knows of a pencil of a sort. Cigarette. Okay, I also have, these days, you don't see too many people smoking around. So probably some people haven't even seen cigarettes, but as for pencils, but so it's a cigar shape sort of thing. And that is what it is. This tapered hair sperm often contain abnormal chromatin or poor packaging of the DNA genetic material. So in all of this, where the head is abnormal, it's likely to have an abnormal DNA or genetic material, and therefore also likely to have an abnormal acrosome, and therefore likely to have fertility challenge. Okay, all right. Then thin, narrow is also a variation of the tapered. All right. They are seldom identified and may be due to broken DNA, varicocele structure. So whenever you have a head abnormality, I mean, the important thing to note is that the head was likely to have an abnormal DNA and also likely to have an abnormal acrosome. And that is where head defects have a problem with fertility. Okay, whether it is pinhead or macrocephalic. Remember, if it's macrocephalic, sometimes it means that, remember we said that the DNA in the process of spermiogenesis, during the transformation, remember the terminology, this is not spermatogenesis, spermiogenesis. That's the stage where the sperms undergo maturation. And we said that apart from the nucleus become eccentric, it has to pack, it has to condense, it has to become smaller. If the condensation, there's a problem, so that the condensation is improper, it is loose, then it's like when you have macrocephaly in humans, if you have a child with a big head, it's also macrocephaly. Sometimes due to a large brain, if the brain itself is large, for whatever reason, it could be due to a tumor, it could be, if the brain is large, now of course it's going to enlarge the entire skull and then makes the head also big. So if in this particular case, if you expect during the sperm development, we expect the DNA to be condensed, to become highly packed. So if it becomes loose, then technically it makes the head also large. And so once that has happened, it means that the DNA has also been compromised, and therefore it is also going to have a problem with fertility. So I've said over and over again that head defects, our concern is that it's unlikely to cause fertilization. It's unlikely to be able to cause a female egg to form a zygote, and for that matter, to become fertilized. So that's where head defects are always taken very seriously, because all of them, whether macro or micro or taper or whatever, all of them will have an impact on the DNA. All right. We've said this before and it's coming back. Head defects, the global zoospermia. And of course, you know that. And that will lack an acrosome, and we explained that already, because we said the acrosome should be linked to the DNA, the genetic material, by a certain protein, the DPY. I'm sure you remember this one. Now, if it is not there, then it means that the acrosome will not form at all. I mean that because it will not be in paste. The acrosome, which is coming from is supposed to surround it. And once it has surrounded the nucleus, it has to be linked. There must be a protein to link it. If that protein is not there, it means that the acrosome will not be in place. And therefore, it will just floating around and then cause the entire head to be rounded up. All right. So if you also have a lot of the sperm, like we talk about the macrocephaly sperm syndrome, if you have a lot of them exhibiting the global zoospermia, you call it total globospermia syndrome. All right, total globospermia syndrome. A variation of this type of sperm is the decondensing head. You see, remember we said that the nucleus, which is going to, from the beginning, which was central, has moved to eccentric, should be condensed. Nuclear condensation is a key process in sperm maturation. Should be condensed. All right. And once it's condensed, it becomes smaller. Then the Golgi body, which is the acrosome, will have to cap it so that it keeps it in shape. Now, with this particular case, global display, we know that there's a defect. And so the acrosome is not there. And therefore, the condensed nucleus become decondensed. It's like it's no longer what condensed. So removal of the condensation. So instead of it to pack, it is now losing out. So it has become decondensed. And so, and of course, it will have no acrosome. And so you see that global spermia will give you a decondensed medicine. And you know that whenever you have a nuclear staining, the nuclear staining will have a unique characteristics. All right. And so once you have the nucleus rather decondensed, you like now instead of impact, it's like spread out all within the head. Then it means that if you do a nuclear staining, the entire head will show that the whole head has the nuclear staining, and that you really know that there's no acrosome even in there. The entire head is made up of the whole nucleus. I hope that you've gotten the point. So that's decondensed, and so it is global spermia and decondensed. Whenever you see decondensed head after staining, you know that it is an example of a global spermia. And when you have a lot of them, just as we have with the microcephalic sperm syndrome, if you have a lot of them, not that you have one or two occasional global spermia and then you say total. No, total means total, a lot, almost all of them are round-headed sperms. And then once you also stain them, you see the entire head exhibiting the nuclear staining, the entire head. And so you say these are decondensed head. All right. And tailless and decondensed. So on the right side, you are going to see the decondensing head. You see that you see them rounded. You see them rounded, and you see the entire head. Okay, doesn't give a space for the acrosome. You see, the nuclear DNA will stain deeply. Okay, and so you are going to see a deep. For example, even if you look here, you see that this one is not a decondensed head because this side is staining the nuclear, and then this side of the head is free. So this is like a normal head. Okay, but almost all of them have this, the whole entire space of the head is staining the nuclear, taking up the nuclear stains. So this could be classified as decondensing head. All right, decondensing head. And then over here, you're talking about tailless. Tailless, you are seeing the head and there's no tail to it. All right. So it's also a form of a head defect. Because the head is supposed to carry, you can't say that this is a tail defect. This cannot be a tail defect because the tail is not even there at all. It is the head that's supposed to have a tail. Okay, so if you have a head without a tail, it's a tail defect. It is not sorry, it's a head defect and not a tail defect. Please take note. Tailless sperm is a head defect and not a tail defect. All right, you have a head which is supposed to have a tail, which it